Now that we discussed what the structure of the skeletal muscle is and what a sarcomere is, let's discuss how the muscle contraction actually takes place within our skeletal muscle. And let's begin with the generation of an action potential. So before we actually contract our skeletal muscle, the brain has to create that electrical signal in the form of an action potential. So it creates the action potential and it sends the action potential down to our somatic nervous system. And the somatic nervous system contains motor neurons, and these motor neurons connect to our skeletal muscle. So basically these motor neurons pick up the electrical signals that are produced by our brain, and they carry these electrical signals in the form of an action potential down their axon and onto the axon terminal that connects to the cell membrane of our muscle cell. So this is our muscle cell, also known as a muscle fiber, also known as the myocyte. And the muscle cell is wrapped around with a special membrane known as our sarcolemma. Now the sarcolemma consists of these invaginations, these tunnels that basically go deep into the cell, and these are known as t-tubules or transverse tubules. They are shown in the diagram with these white dots. So this is the axon of the motor neuron that is carrying our action potential. And essentially the action potential travels along the axon until it gets to our axon terminal. So this is one axon terminal, a second axon terminal, a third axon terminal. Now if we zoom in on this axon terminal, we basically get the following diagram. This is the axon terminal of this end region of the motor neuron, and this is the membrane that is shown in red. So basically once the action potential arrives onto the axon terminal will release synaptic vesicles that carry a special type of neurotransmitter known as acetylcholine. So once acetylcholine is released into this synaptic cleft, these acetylcholines are shown by these black dots. They basically bind to the membrane proteins on our sarcolemma, and that will depolarize our sarcolemma. Now, once the sarcolemma depolarizes, that action potential that is created on the sarcolemma will travel into the cell via these deep invaginations known as our t-tubules. So what these t-tubules basically do is they allow the action potential to very quickly and evenly spread throughout the entire muscle cell. So once our action potential travels into the cell, it will basically cause the activation of these channels found on the membrane of the sarcoplasmic reticulum. So remember, the sarcoplasmic reticulum is basically the specialized type of endoplasmic reticulum found inside our muscle cell. So if we take a cross section of our muscle cell, the green region is basically our sarcoplasmic reticulum. So this is our sarcoplasmic reticulum. Once it accepts that action potential, once the action potential travels to our sarcolemma, it will basically open up these special channels on the membrane that will basically allow calcium ions to flow into our cytoplasm. Remember, the cytoplasm of our muscle cell is also known as our sarcoplasm. So inside our sarcoplasmic reticulum, we have a high concentration of calcium. On the outside, in the cytosol, we have a low concentration. So these calcium ions will travel down their electrochemical gradient and into our cytosol. Now, inside the muscle cell, we have many of these myofibrils. And what these myofibrils are basically our sarcomeres connected end to end. So we have many of these myofibrils, and if we examine one of these myofibrils and we zoom in on the myofibril, we basically get the following diagram. So we have many of these sarcomeres connected end to form our myofibril. Now, once the calcium is released into our surroundings, into the cytoplasm of the cell, they will go on, the calcium ions will go on and bind to a special region on the thin filament, as we'll see in just a moment. Now, by the way, this junction between the axon terminal and the membrane of our muscle cell is known as the neuromuscular junction, and it's also sometimes known as the motor end plate. So if you ever hear the expression motor end plate, that simply is referring to the synapse between our end of the motor neuron and our sarcoplasm of our muscle cell. This is also known as the neuromuscular junction. So now that we know how the signal actually arrives onto our muscle cell and what it causes, let's see what the calcium actually does and how it interacts with the thin and thick filament. So this is our sarcomere. The sarcomere consists of these thin filaments that are composed of globular proteins known as actin. And we also have the thick filament shown in purple that are composed of our protein known as myosin. So let's discuss how they actually interact. So let's label this step as step number 1. So basically the release of our calcium into the cytoplasm, into the cytosol of the cell, that is step number 1. Now, what about step number 2? Step number 2 is the binding of that calcium to a special protein found on our actin filament, on the thin filament, known as troponin. So we have this protein that is shown by these 3 globular pink regions. This is troponin. A tropomyosin basically is this other blue protein that extends all the way around our actin filament. And our troponin is actually bound to our tropomyosin. So as soon as calcium binds to our troponin, the troponin basically changes its shape. It experiences a conformational change. And as it changes its shape, it causes our tropomyosin to also change its shape and shift. And as our tropomyosin, shown in blue, actually shifts, it exposes special binding sites on the actin filament known as our myosin-binding site. So this is basically where our myosin heads found on the thick filament actually bind to our thin filament. So this green region, which was blocked on this diagram, is exposed as soon as our calcium binds onto our troponin. So this is our calcium, it binds onto our troponin, it changes its shape, which in turn changes the shape of our tropomyosin, and that exposes our myosin binding site. So this is step number 2. Now, as soon as our binding site is exposed, right before the binding between the thick and the thin filament actually takes place, our myosin head has to basically hydrolyze an ATP molecule. So the myosin heads contain ATP molecules, or an ATP molecule, so 1 ATP per our myosin head. Now, before they can actually bind to the myosin binding site that was just exposed as a result of the binding of the calcium, the ATP must be hydrolyzed into ADP and a phosphate group. So basically, this ATP that is found on the myosin head of our thick filament, so this is the thick filament, this is our myosin head, which is basically our myosin heads on this thick filament shown here. It is hydrolyzed, and the ADP and the P that are produced remain on that myosin head. So this is step 3. Now, once we hydrolyze the ATP into ADP and the phosphate, and once the calcium binds onto our troponin, exposing that binding site, now the binding can actually take place. So now the myosin head is oriented correctly. So what also happens when we have our hydrolysis is, in this diagram, the myosin head is oriented at a 45-degree angle with respect to our thick filament. But when we have hydrolysis taking place, when we produce the ADP and the P, this myosin head orients itself approximately at a 90-degree angle with respect to our thick filament, as shown in this diagram. And now the orientation is just right for this myosin head to actually bind to our binding site, as shown in this diagram. And this binding, let's label this as step number 4. Now, what takes place once they actually bind? So once the thick filament and our thin filament actually bind to each other as a result of our myosin head, step number 5 takes place. In step number 5, the phosphate group and the ADP group are basically released from our myosin head. And as this is released, that causes the change in orientation of the myosin head. The myosin head once again goes from a 90-degree angle to about a 45-degree angle. And as that takes place, that moves our thin filaments towards each other. So if this is one portion of the thin filament, this is a second portion of the thin filament of the sarcomere, they basically are pulled towards each other. So these Z lines, this Z line number 1 and this Z line number 2, basically are pulled towards each other. And so we have this motion taking place. This entire thin filament basically is pulled this way as our ADP and P basically detach from the myosin head, and the myosin head is oriented once again at the 45-degree angle. So once it binds, once the myosin head binds onto the binding site, it must expel the ADP and the phosphate molecules in order to actually move the myosin head oriented at an angle of 45 degrees with respect to our thick filament, and that essentially pulls the thin filament as shown in this diagram. So this thin filament basically is pulled this way so that these two Z lines are basically pulled inward. Now, this process of actually pulling on our thin filament by the movement of our myosin head is known as the power stroke. And the power stroke must take place when our phosphate is basically expelled from that myosin head. So once that power stroke actually takes place, we have to detach that myosin head from our thin filament. And the way that we do that is we basically bind an ATP molecule onto our myosin head. So in the last step, we basically take an ATP molecule and we bind it onto our myosin head, and that releases our thin filament from our thick filament. At the same time, the calcium that is bound to our troponin is basically released. And that means our tropomyosin closes that binding site. So now our myosin head can no longer bind to our actin filament. So when ATP binds to our myosin head, it causes it to detach. Calcium also detaches, thereby blocking those myosin binding sites. And this concludes our contraction of the muscle. And this process basically repeats itself. So essentially, when I'm moving my skeletal muscle back and forth, this process is taken over and over and over.